You can have two pills with the exact same amount of active medication, but while one relieves your symptoms in 20 minutes, the other might take hours to even start working. The difference comes down to a process called dissolution, the specific rate at which a pill breaks down and releases its payload into the body. For generic drug manufacturers, this presents a massive challenge. They cannot simply copy the ingredients of an expensive original drug. They have to mathematically prove their version releases the medication at the exact same rate. If the generic's dissolution profile doesn't perfectly mirror the reference drug, the patient's absorption is thrown off, directly compromising their treatment and safety. Let's look at how this works in practice. Imagine a pharmaceutical company has formulated a new generic immediate-release tablet and needs to test it against the brand name reference. To test the physical performance, both the reference and the test pills are dropped into simulated bodily environments. Scientists then extract samples at specific intervals and record the percentage of the drug released at 5, 10, 15, 20, and 30 minutes. This chart plots the percentage of the drug released over time. To the naked eye, the two release curves look almost identical, but regulatory agencies demand strict numerical proof, not visual estimates. Visual similarity isn't enough to pass quality control. The data must be subjected to two strict mathematical stress tests. The first tool is called the difference factor, or F1. It calculates the relative error between the two products. We start by setting up a data table that aligns the reference and test percentages. Right away, we can see the turmeric is releasing several percentage points less drug than the reference during those first 5 and 10-minute intervals. Next, we add those individual differences together to calculate a total sum of deviations. Finally, we divide that sum by the total reference dissolution and multiply by 100. For our generic case study, this leaves a final F1 score of 22. The regulatory acceptance criteria for F1 is a score of 15 or lower. Because 22 is greater than 15, the generic has failed the first test of similarity, proving that the physical gap in that critical early release window is simply too wide for quality control to ignore. There is a heavier, more heavily weighted statistical tool preferred by international regulators, known as the similarity factor, or F2. Before F2 can even be calculated, the testing must survive a rigid gauntlet of conditions, starting with testing a required batch of exactly 12 dosage units under perfectly identical and controlled laboratory environments. The strictest rule demands that only one sampling point is allowed to be above 85% drug release. Global health authorities require these rigid prerequisites to ensure that random experimental noise doesn't manipulate the data. This formula highlights how F2 operates. By squaring the differences, the calculation severely punishes large deviations, like the ones at the start of our test. The generic's calculation results in a score of 42. This falls completely outside the acceptable minimum of 50. Failing the F2 score means the generic formulation is entirely rejected by regulators and sent all the way back to the drawing board. The formulation team uses the failed F1 and F2 scores as a diagnostic tool. Since the math confirms the tablet is lagging during those initial minutes, they have a clear lead on the physical problem. This translates to a physical manufacturing issue. The team used an excessive binder concentration and compressed the powder too tightly. They physically optimized the pill by reducing the binder concentration, introducing a super disintegrant, and lowering the machine's compression force. Abstract statistical equations directly dictate the physical engineering of the medicine. The newly engineered tablets are sent back to the lab for a completely new dissolution study. This dashboard displays the new scores. The optimized formulation achieves an F1 of 4.3 and an F2 of 78. With these passing numbers, the generic product is proven to match the reference. It is formally cleared for scale-up and regulatory submission. Similarity factors are the essential mathematical bridge guaranteeing a cheaper generic is exactly as effective as the original drug.